preview session. After we go to the Q&A, if there are learning objectives. And then at 9 o'clock, or excuse me, 9:50, we'll call it quits. And we'll get out of here so that there's room for genetics. So, guys, girls, any questions? Sure thing, we could definitely review that. Let's pull up the pictures. So when we think of a communicating rami, I just want you to think of a branch that helps to connect the sympathetic trunk ganglion to the rest of the spinal column. So autonomic. We'll start up right here. So before we talk about rami, let's just reinforce the sympathetic chain ganglion or sympathetic trunk ganglion. Lateral to the spinal column on the left and right sides, we have a bunch of ganglion. And in this context, ganglion is a bunch of cell bodies of neurons in the peripheral nervous system. I circled them in red in this diagram earlier. Let me clear the picture for us. So we have the ganglia, which are the bulges. And then those ganglia are going to be connected by nerves. Those nerves that are going to connect the ganglia are called rami, sometimes also called communicating rami. And as we look at those rami, or communicating rami, of the sympathetic chain ganglia, there's a couple different paths of travel that the signal can take. Regardless of the path of travel, we're going to have some general terminology. We have a preganglionic fiber, a postganglionic fiber. So when we talk about a preganglionic fiber, this preganglionic fiber is before the ganglion, hence the suffix pre. So it's going to go directly from the spinal nerve to the ganglion. With our postganglionic fibers, they are after the ganglion, hence the suffix post. These are going to be fibers that leave the ganglion and carry that action potential to the rest of the body. As we look at postganglionic fibers will always go to the organ. This is a little bit of background associated with the rami. Depending on the branch of the autonomic nervous system, sympathetic versus parasympathetic. Our sympathetic branch of the autonomic nervous system has ganglia very close to the spinal column. So our sympathetic branch has a short preganglionic fiber and a long postganglionic fiber. And then when we look at the parasympathetic branch of the autonomic nervous system, the parasympathetic branch of the autonomic nervous system has a very long preganglionic fiber. The ganglion is nearly at the target organ, and then the postganglionic fiber is super duper short with the parasympathetic branch of the autonomic nervous system. Did you want to talk about the three methods of travel within the... All right, let's talk about the three methods of travel. Those three methods of travel are numbered up on the screen for us. We have the one, let me circle it in a different color for us. Having green next to green is really hard to see. We'll go with purple. Let's do purple. So we have the 1, and then 2 as superior and inferior, and then 3 to the splenic nerve. So our first way is that some of those fibers are going to end up in the ganglion, and then they're going to synapse and then immediately go to the postganglionic organ. So that's one potential method of travel. Let's back up a slide. In other words, the preganglionic fiber goes directly to the ganglion, and then from the ganglion, the postganglionic fiber goes directly to the target organ. That's technique or path of travel number 1. That's the short, sweet, most direct path of travel for the sympathetic nervous system. Then we have method number 2. For method number 2, we're going to have that fiber up or down. So we're going to have the fiber synapse at a ganglion at different levels within the sympathetic trunk ganglion. So let's back up a slide here. And we'll clear our diagram. So we can have the preganglionic fiber go from the lateral horn of the gray matter through the spinal nerve to the first ganglion, and then it'll either head superiorly or inferiorly and synapse at a separate ganglion that's superior or inferior to the initial ganglion that it entered. From there, after synapsing, the postganglionic fiber will travel directly to the target organ. And then we had method number 3. For method number 3, that ganglionic fiber is going directly through the chain and not synapse at all with any of the ganglion in the sympathetic chain ganglion. Instead, we'll have it form a splenic nerve. And in this situation, for scenario number 3, the preganglionic fiber goes through the anterior root, goes through the spinal nerve, travels directly through the sympathetic chain ganglion, and then will synapse at a secondary ganglion separate from the sympathetic chain ganglion. And as it's doing that, there will be a bundle of axons referred to as a splenic nerve carrying it away. That is the most complicated of the 3 options. So in terms of complicated, or in terms of difficulty of travel, method number 1 is the simplest and most direct. Method number 2 is slightly more complicated. And then method number 3, forming a separate splenic nerve, is the most complicated. In the back. It doesn't synapse at the ganglion. So the difference between a preganglionic and postganglionic fiber is the separation of the physical separation of the neurons. So when we go from the first neuron to the second neuron, that separates the pre versus post ganglionic fibers. And to have that transition from the first to the second neuron, we have to have a synapse occur. So when we think of the splenic nerve, let me clear our screen again for us. That preganglionic fiber starts in the lateral horn of the gray matter and passes directly through the ganglion without synapsing, and then we'll synapse in a secondary ganglion. What was that? The part that I traced in purple is all preganglionic fiber. Excellent questions. Any other questions related either to ganglion fibers or just the nervous system in general?
So I guess I'm just having a hard time understanding. Is the autonomic nervous system like not part of the CNS or CNS, like it's its own section? Like I guess like how do those relate to each other?
Good question. Let's go, I think, the second slide that we covered on the nervous system. Maybe it's the 4th or 5th slide. Nervous system overview. So our autonomic nervous system is also known as the visceral motor division, which is a subcategory of the peripheral nervous system. So when we think of the autonomic nervous system, we really dive into the details. This arbitrary flowchart that I gave you the very first day we started covering the nervous system doesn't necessarily do it justice because there's some gray lines. It's not as black and white as this chart leads you to believe. Because when we think of the autonomic nervous system, that preganglionic fiber starts in the spinal cord, starts in the CNS, but then travels to the PNS, exits the spinal cord. So that's the autonomic nervous system is one of those branches of the peripheral nervous system that's partially in the central nervous system. But because the vast majority of its tissues are outside of the central nervous system, we categorize it as a PNS function. Any other questions, comments, concerns? Well, I have something I'd like to tell you. I didn't want to spam you with a lot of, I've been sending lots and lots of course announcements this last week, and I didn't want to send a separate course announcement to let you know that I had finalized the breakdown for the exams. So I just added it to a previous announcement I posted. For those of you who plan on taking only the Unit 4 exam, we have the Unit 4 exam breakdown available for you to view. The Unit 4 exam is going to consist of 50 total questions. Of that, 15 will be from the nervous tissue, 5 will be on the spinal cord, 15 will be on the brain, 5 will be on the autonomic nervous system, and 10 will be on the ear and eye for the Unit 4 only version of the test. For those of you who plan on taking the cumulative version, you have all of the Unit 4 questions plus 50 cumulative questions. The 50 cumulative questions, open with photos, always, includes everything else that we covered in the class. So we're going to have 4 questions on the introduction to the anatomy and physiology. We'll have 4 questions based on chemistry, 11 questions based on molecular biology. And I was trying to find a good heading for molecular biology. That includes mitosis, the DNA replication, protein synthesis, cell cycle, cellular respiration, all those like there's, you know, we spent a good chunk of time on all those topics. So 11 questions based on those topics. 5 questions on histology, 4 on tegumentary, 9 on skeletal, 7 on muscular, 2 on joints, and 5 on endocrine. So that's the breakdown of the cumulative portion. And I'm done writing it, and I can assure you these are big picture questions for the cumulative portion. We'll go at the back and then in the middle. The location of the limbic system. The limbic system is part of our cerebrum and partially diencephalon as well. As we look at our limbic system, it is, let me pull up the picture for us. Sometimes this website gets a little glitchy on me. Here we go. Cerebellum and cerebrum. I believe I covered limbic system in this presentation. Language, nope, it was at the end of the preceding presentation. So let's pull up our diagram. Limbic system, just to reemphasize this, has a functional categorization than a structural categorization. The structures of the limbic system serve dual purposes. They're part of the limbic system, and then they're of another function or another system within the brain. When we think of the limbic system, I want you to think of strong emotions and learning. I've heard the limbic system described as sex, sleep, and satiation before. Just very primitive functions associated with strong emotions. As we look at our limbic system and the structures involved with the limbic system, some of those key structures include, let's shift back to red here, the hippocampus, which is involved with taking short-term memory and converting to long-term memory. And then we'll have our amygdala, directly rostral or anterior to the hippocampus. We'll also have the cingulate gyrus. Our cingulate gyrus is right here, directly superior or superficial to the corpus callosum. And as we look at the limbic system, there are some other secondary structures that I didn't mention specifically within the PowerPoint. Please don't worry about those structures. If I didn't spell them out specifically in the PowerPoint presentation, I don't expect you to commit them to memory. For instance, I don't expect you to associate the thalamic nuclei with the limbic system, even though it is technically part of the limbic system. So the 3 main structures that we've emphasized with the limbic system are listed on this screen and circled in red. Does that answer your question? All right, there was one in the middle. Question 1 of the Chapter 12 practice quiz. Let me pull it up on the screen for us. Sure thing. Can I get it out? It looks like this is as big as I'm going to get it on the screen. So chapter 12, question 1 states, the nervous system employs exclusively electrical means to send messages, whereas the endocrine system communicates by means of chemical messengers. And this is a false answer. The part that a lot of people overlook is that, yes, the nervous system overwhelmingly uses electricity, but from neuron to neuron, at a chemical synapse, it's going to use a chemical known as a neurotransmitter to send that information. So that's why this is a false statement. The recurrence of synapses?
No, the excitatory coordinates, the inhibitory GABA,
Absolutely. So as we look at our synapses, there are, generally, broadly speaking, excitatory and inhibitory synapses. An excitatory synapse is going to raise the membrane voltage closer to threshold. An inhibitory synapse is going to lower the membrane voltage farther from threshold. It's going to hyperpolarize the membrane. So that's a broad framework to keep in mind. Oh my goodness gracious. There we go. So we want to talk about the different synapses. So let's first talk about the cholinergic synapse. When we think of a cholinergic synapse, our cholinergic synapse is your standard acetylcholine-based synapse. So when we think of the muscular junction that we talked about in a lot of detail, that's a cholinergic synapse. Acetylcholine is released. As that acetylcholine is released, it's going to bind to an acetylcholine receptor. That acetylcholine receptor is then going to allow ions, sodium ions, to rush in. And as sodium ions rush in, that raises the membrane threshold, the action potential. We also have acid or GABA-based synapses. These kinds of synapses are going to rely on GABA as its neurotransmitter, and they are going to open up chloride channels. When we think of a chlorine ion, they're in group 17 on the periodic table, period 3, and they have 7 valence electrons, so they'll have a -1 charge in their ionic state. They're highly electronegative ions that are going to lower the overall membrane voltage as we add more chlorine ions to the intracellular side or intracellular fluid of the membrane. So when GABA binds, it opens up chloride ion channels. When we look at our concentration gradient, there's a higher concentration of chloride outside of the cell. So by opening up those channels, chloride ions will enter the cell and make the inside more negative. When the inside becomes more negative, its voltage moves farther away from threshold, inhibit the generation of an action. Then we have our adrenergic synapses. Adrenergic synapse. This is going to rely on a decoupled protein across the cell membrane. Instead, these molecules will bind to a transmembrane protein on the surface of the cell. And as that name implies, transmembrane proteins go from the extracellular to the intracellular side. They go directly across the cell membrane. And when the epinephrine side of the transmembrane protein, it causes the intracellular side to have a conformational shift or change shape. That changing shape on the intracellular side is going to initiate a protein cascade where we'll take secondary messengers and amplify the signal. The secondary messenger in this particular situation is cyclic AMP. Another common secondary messenger is going to be calcium ions, which are not the particular secondary messenger in this particular situation. As the secondary messengers move throughout the cell, they amplify the signal and try to bind to ion channels. And as they bind to the intracellular side of our ligand-gated or chemically gated ion channels, they can cause sodium ions to rush in and initiate the action potential. So those are the 3 kinds of neurotransmitters. One student in this class was emailing me really detailed questions on the nervous system that forced me to pull up my neurophysiology textbooks. And while I was reviewing for her and picking up the answer for her, I noticed that there was about 300 pages dedicated to neurotransmitters. So right now, we have three or four PowerPoint slides, but keep in mind, this is a really deep topic. We're just barely scratching the surface right now. Any other questions, comments? All right. Oh,
For the functions of each lobe, are we gonna have to know like very specific? Like, I think there was a slide with like a picture and then it had like a list that said like all the functions.
yes, absolutely. No. So I'll pull up that slide so that everyone can know what we're talking about. The answer to your question is no, you don't have to memorize everything on that slide. I believe it's this presentation. Doo-doo-doo. Is this the slide you were referring to? For this slide, guys, gals, please don't memorize everything. In the brain integrative functions PowerPoint, I typed out specific functions associated with specific lobes in specific parts of the brain. Focus on that stuff that I specifically typed out. The purpose of this slide isn't necessarily to overwhelm you, but just to help illustrate that it's the reality, the truth of the situation is much more convoluted. Any given function is not going to be exclusively localized to a given part of the brain. Instead, we find that a given function will have most of its processing associated with a specific part of the brain. But when we look at the functional parts of the brain, we're going to have a lot of overlap, where a single part of the brain is going to do multiple things. Yeah, that's the natural follow-up question. What do I really need to focus on if not this slide? Let me pull up that up for you. That was in brain and integrative functions, where we went slide by slide in this PowerPoint and talked about individual functions and where they are overwhelmingly going to be located within the brain. So when it comes to the brain and functions, focus on the brain integrative functions PowerPoint for you. That's the part that I really want to focus on. So are you asking what is the difference between a rami and a spinal nerve? Or let me see if I can answer your question. I'm not 100% sure what you're asking. So let's go back to the rami. We have spinal cord. Oh, wait, nope, rami was in the autonomic nervous system presentation. Let's pull up that diagram for us. So are you asking what makes a rami a rami? So the communicating rami are just connections between ganglion. The meningeal branch is a specific branch that instead of heading out to the body as a whole, is going to loop back in and innervate the meninges of the spinal cord, which allows for us to perceive pain associated with damage to those meninges. Question. Any other questions, comments? Sure thing. Let's pull up the picture. You may remember a couple weeks ago in lab, we had pyramidal cells on a microscope slide for you to look at. Those pyramidal cells are going to function by connecting the surface of the cerebral cortex, the neocortex, with the deeper parts of the brain, to the ganglia, deeper into the cerebrum. Cerebral cortex, here we go. Pyramidal cells. So when we think of the cerebral cortex, just for the background, the cerebral cortex is the surface of the cerebrum, or the gray matter on the surface of the brain that forms the sulci and the gyri. This is where we have most of our gray matter. Our cerebral cortex in human beings can also be referred to as the neocortex. Most organisms don't have this hierarchy of structures. And when we look at it in a human being, we find there are multiple layers to it. We start out with gray matter on the surface, and that gray matter has a very high concentration of dendrites and neurosoma, or cell bodies. And then we'll have axons carrying the information away, or we have lots of myelinated fibers. Most of those myelinated fibers that are carrying that information away from the cerebral cortex, or the surface of the cerebrum, are going to be attached to a specific kind of neuron called a pyramidal cell. You could think of a pyramidal cell as a tetrahedron, those of you who had to memorize geometric shapes within your classes. So a four-sided pyramid, 3 sides going up, 1 side on the bottom. And this triangular pyramidal cell is going to have very long dendrites that synapse with the cells up in the gray matter, and then it's going to have a very long axon that will carry that action potential to the nuclei, or the regions of gray matter deep within the cerebral cortex, or cerebrum. Did that answer your question? Awesome. We'll go in black and then in gray. Good question. Talking about the cerebrum, which is part of the brain and is absolutely part of the central nervous system. Within the peripheral nervous system, we have neurons. So you stand for just a multipolar neuron. It's going to be the most common part of the most common neuron of the peripheral nervous system. Today during class, when we talked about vision, we talked about bipolar neurons, and we also talked about ganglionic neurons. Those are specialized neurons that function with special senses. But when we think of the most common kinds of cells within the peripheral nervous system related to the nervous system, we have multipolar neurons, and then we have Schwann cells and satellite cells, where the Schwann cells will myelinate the axon. With that, it at the spinal column, because low down in the spinal column, where we have the cauda equina, we can insert the needle and not worry about directly puncturing or damaging the specific tissues or nerve fibers of the central nervous system. Within the brain, we don't have those fibers that can move out of the way, so we typically don't try anesthesia. The epidural space within the brain. Our epidural space within the brain is, depending on who you ask, either filled with collagen fibers carrying it to the or it's going to be just a very, very small amount of So the epidural space is the space outside or superficial to the dura mater, and within the brain, the epidural space is dense bone tissue of the cranial plates. And occasionally, you'll see that pull away and have a little bit of fluid build up in it. Most of the time, it's going to be filled with compact bone tissue. You need to know what we covered in class. So if you check the lecture textbook, there's a lot more detail in the lecture textbook. And if you start Googling stuff, you'll be overwhelmed very, very quickly. So focus on what we covered in class in the PowerPoint. That's what you need to know for the cranial nerves. De-innervation hypersensitivity was when I had a really bad analogy during class that crashed and burned. I remember specifically talking about how bad that analogy was afterwards. With our autonomic nervous system. Our autonomic nervous system has two branches, the parasympathetic and the sympathetic branch, that help to upregulate and downregulate different parts of our bodies. You could think of the autonomic nervous system as fine-tuning part of our nervous system. It helps to take the reins of the activation of our organs. Without the autonomic nervous system, you either be all the way on, all the way off with most of our visceral organs. But our autonomic nervous system allows us to have partial activation. And if we don't have the autonomic nervous system allow for us to have partial activation, or that fine-tuned regulation of our visceral organs, we are going to have hypersensitivity, de-innervation hypersensitivity. The first word, de-innervation, refers to severing the nerve connection. And then hypersensitivity refers to the fact that we are going to have an overstimulated or overregulated activation of the visceral organ. I think the bad analogy I used in class started with a carburetor, and then I switched to a candle on a bonfire, and it just didn't work. Let's focus on the generals. So, what versus but then in particular with the left hemisphere, we talked about Wernicke's area, we talked about Broca's area, and then within the right hemisphere, we talked about the ability to perceive tone or understand sarcasm. But these are generalizations. It's worth emphasizing that children are much less rigid in their hemispherical designations. They have a lot more neuroplasticity. Adults are much more rigid. It's harder for us to rewire our brains. And the adults, males have much more lateralization. Males tend to be more right brain versus left brain or vice versa. In males, one brain will dominate more than the other one typically. And in female brains, where the corpus callosum is proportionally much larger than it is in male brains, we find that female brains, generally speaking, don't have one side dominating over the other side as often as it happens in male brains. So females are better between the intuitive, artistic, emotional and the recognition side of their brains with more ease, we should say, compared to males. Or according to my wife, because females are smarter. Any other questions, comments before I start diving into our learning objectives? All right, let's just dive into the learning objectives then. Let's see here. Dropbox is where I have it. My files, 214.